Our next speaker is Professor Hikoya Hayatsu from Okayama University. Hikoya, I have known him for a long time, ever since I've been in Korea's lab. And the thing is that he has always been afraid in me. It's a in-joke. Okay, Hikoya. Can I start? Yeah.
Thank you.
All right, ladies and gentlemen. I came here, Madison, 45 years ago as a postdoc to Oregon Corona. I came here with my wife, sitting somewhere behind, and our first son, only half a year old at the time. And we spent 2.5 years between 64 and 67. Today I'm talking about the bisulfite modification of nucleotides. Since I went back to Japan after finishing this genetic code related synthetic work, synthesis of 64 triplets, just like just the ones last night Dr. Nierenberg talked. I was one of the four-man team to synthesize 64 rival triplets. Now, coming back home, I saw that maybe I prefer not synthesis, but breaking down DNA. So, I focused to find some ways, some new ways to modify nucleotides and nucleic acids. And I'm today talking about bisulfite modification to be used for the DNA methylation analysis. So this simply shows the major four bases of DNA: thymine and cytosine, pyrimidine bases, adenine and guanine, purine bases. Now, in addition to these four bases, there are modified bases in DNA discovered. The 1,5-methylcytosine. This is a major subject of my talk today. Was discovered in calf thymus DNA by Wyatt in 1951. Wyatt is a British, I believe. And then 5-hydroxymethylcytosine. This is oxygenated here. This was discovered in T2, T4, and T6 phases. In 1953, again by Wyatt. It is remarkable that very recently this base was discovered in animal DNA. In mouse brain, these two scientists reported the presence of 5-hydroxymethylcytosine, reported in the April issue of Science this year. 6-methyladenine, 6-methyladenine discovered in bacteria, again by Wyatt. This book, The Modified Nucleotides, was written by Ross Hall, published in 1971. Ross used to be a colleague of Gordon in the 1950s and in UBC, Vancouver. And I was told by Tom that he recently passed away, unfortunately. This book has been very useful for me, working on the modified nucleotides in DNA. Now, in this book, he wrote 40 years ago that he isolated 25 grams of DNA from mouse and digested it into nucleosides and tried to fractionate and see whether any odd nucleotide side can be found or not. And he said only 5-methyl-deoxycycline was found. And he also said that probably no other minor bases of labile nature, except for the labile nature, would be present in this animal DNA. But it turned out to be wrong. As I said, as I told you in the previous slide, we have 5-hydroxy derivative of cytosine, methyl derivative of cytosine, was indeed found in a mouse brain, and it could be very important in the future biological work. Five-methyl-cytosine, 5 here, 6 here. Animal DNAs contain 5-methyl-cytosine up to 10% of cytosine. Plant DNA somehow up to 30% of cytosine. Very large amount. This base pairs with guanine, just like cytosine. So it behaves like cytosine, and by ordinary DNA sequencing, you cannot tell the location of this modified base in your sample. This base has been regarded as not mutagenic because it pairs with C and G, just like C. But recently it was almost established that in the sunlight-derived skin cancer, mutations occur through the mediation of this 5-methylated derivative. There's a lot of arguments and papers recently going on, but I do not go into that. 5-methyl-cytosines are present primarily in CpG science. Now, how 5-methyl-cytosine works? Gene silencing is a hallmark of methylated cytosines in DNA. This is a sample in a human mental disease called Fragile X syndrome. In this patient, many cytosines in this promoter region of this codon or a protein, many cytosines are methylated. Whereas in a normal human being, they are not methylated. Now, in this mental retardation disease, this methylation makes the coding region not transcribable. Transcription does not take place in a normal way. So the result is greatly decreased production of the protein, which is necessary for nerve functions. Now, I took this from this very nice textbook, Understanding DNA. 5-methyl-cytosine-induced effect like this is called epigenetic effect. It is now quite well known that epigenetic effect is connected to cancer, related to cancer, and also to aging. Many cancer tumor suppressing genes may be silenced by the hypermethylation of cytosines so that the tumor can freely grow. In the aging, methylation is now thought to be related to the difference of identical twins. Identical twins, one of them may get cancer in their old age, very probably through the different patterns of methylation between these two identical twins. Now, 5-methylcytosine in the genome can be analyzed by bisulfite genomic sequencing. Bisulfite genomic sequencing is a very popular word now. But in 1970, when I started my work Japanese research in Japan, in Tokyo University, at that time 5-methylcytosine, its presence was known, but its biological role was entirely unknown. But in a very rapid chance, we discovered that bisulfite, a very common reagent, laboratory reagent, anybody can use that. Bisulfite can mediate the amination of cytosine and 5-methylcytosine as well. Both cytosine and uracil, both are these two pyrimidines, uracil being the major component in RNA, thymine in place of thymine in DNA. Bisulfite adds across this double bond, 5-6 double bond of these pyrimidines to form this atom, 5-6 dihydroatom. Cytosine in DNA should be very stable, otherwise we die very easily. But this amino group is chemically very stable in aqueous solution. But once this double bond is saturated, this amino group becomes very labile, so that in the aqueous solution it spontaneously deaminates to give the uracil bisulfite compound. And this uracil bisulfite adduct can be converted to uracil simply by treatment with alkaline. These two steps, step 1, step 2, this occurs simultaneously in a one-pot experiment. The pH required for this reaction is acidic pH. pH 5 to 6 is an optimum. And similar reaction occurs for 5-methyl-C, but the difference is the speed of the reaction. This is relatively fast, but 2 orders of magnitude smaller rate was observed for 5-methylcytosine in our experiment at that time. So at that time, I did not realize that this has turned out to be very, very important today to discriminate between cytosine and methylcytosine. Now, in 1980s, the biological significance of 5-methylcytosine became apparent, and scientists suddenly began to need the discrimination methodology to discriminate between 5-methylcytosine and cytosine. And in 1992, a group of Australian scientists devised a very nice means to differentiate between cytosine and methylcytosine. Bisulfite converts cytosine to uracil, as I told you. But methylated cytosine resists to this treatment. So in some given conditions, no reaction takes place. So for a sample containing cytosines, bisulfite treatment would give uracil. Whereas for the same sequence but methylated strand would give no conversion. This stays on. So using PCR, polymerase chain reaction amplification, you get descendant group of DNA molecules having T here instead of the original C. Whereas in this case, CM, as it behaves like C, the result is the production of many molecules having C. So by knowing and comparing these sequences with the original sample of your DNA, you can tell between C and C and methylated C in this way. So since that time, this original protocol has been taken up by many scientists, and epigenetic studies have been done mainly using this technology, bisulfite genomic sequencing. 1 week ago, before I left Japan, I checked internet with a keyword bisulfite sequencing. I found 117 papers. This is 6 years, almost one paper in 3 days, increasing. Very, very active field. So I'm quite happy now that our original discovery in 1970 has been taken up by people and used for such very important studies. The studies include not only diseases, cancer and other diseases, related diseases, also for aging. So in the very future, people predict that we will have methylation imaging map of human beings. So this technology is very important. And in a textbook, conditions generally used for the bisulfite treatment was like this: 5 molar sodium bisulfite, pH 5, 50 degrees, 4 to 16 hours. 5 molar sodium bisulfite, in my experience, is the saturation level. You cannot go up more than 5 molar with sodium bisulfite. Please note that this length of incubation is rather long. So, several years ago, I started to wonder whether I can contribute to this field by improving the protocol. Particularly, if I can make a speeding up of the whole procedure, it would be useful. I knew from our old publication that the rate bisulfite concentration ratio was like this. It is very strongly, this rate is very strongly dependent on the concentration of bisulfite. It is more than linearly related. So increasing the bisulfite concentration more than 3 molar, you could expect very high speed of the reaction. So this line here, these points are experimentally observed points. The line shown here was drawn on the assumption that bisulfite participates not only in this addition process, but also somehow in this deamination. So 2 molecules of bisulfite is seen to be participating. Otherwise, this line would become this way. So we somehow this participates. The mechanism is still unknown. So this is a very important thing I have to work on until my age of 100. Hopefully. So my greatest hobby is to do experimentation on the bench. I still am doing. Now I tried 4 years ago. I tried to make 10 molar bisulfite instead of 5 molar. In order to make it, I knew in my old experience that ammonium bisulfite is more soluble. So ammonium bisulfite plus some sodium, I was able to produce 10 molar bisulfite. Great triumph. I was very happy. And at the same time, I devised very simple means to quantify bisulfite in solutions. People using these days, these 100 paper people They do not quantify bisulfite. They use just kits, commercially available kits. No, they do not doubt about the concentration. But I always doubt anything like kits. So I do the concentration quantification all the time. Now, anyhow, so I'm quite sure that this is 10 molar. Now, we treated first bulk DNA, some testes. Now, 9 molar bisulfite, 90 degrees, only 10 minutes. Nucleotides on HPLC show that cytosine was completely 100% converted to uracil in a quantitative way. 5-methyl-cytosine very nicely remained as such, no decrease. So it looks like very, very quick reaction can take place on DNA. So we took several standard genomic DNA samples on which methylated positions are known and tried this very nice looking protocol. And overall speed up was like this. 9 molar bisulfite. Original 10 molar bisulfite was mixed with 1/10 of volume of DNA solution. So it is now 9 molar. 9 molar, 70 degrees, just for 40 minutes. Gave very satisfactory results. And we published it in 2004. In the current protocol published by Wiley, I wrote the overview of the whole thing. Now comes the evaluation from other laboratories. This one by Chiba and Sasaki of the National Institute of Genetics, Mishima, Japan. They took mouse gene unmethylated and methylated. And they used 8 molar bisulfite, 70 degrees, 60 degrees. And this shows many clones, sequences in CPG sites, in different CPG sites. White circles show the correct answers. The single black one was wrong, false. So they are quite nice. They get quite nice results. With the methylated genes, there are several false answers, but still they have been very, very happy using this new method protocol. In a recent issue of Nucleic Acid Research, published in December last year, these people from the United States, Seattle, University of Washington, compared conventional procedure with our new high concentration quick procedure using their synthetic oligonucleotides, wherein methylated sites are defined multiple positions, no methylated sites are defined positions, and made this error frequency table. With the conventional procedure, failed C2U conversion, 6%, inappropriate conversion of 5-methyl C to T. This is a conversion unwanted, 3%. In total, about 10% with this conventional method. With our new procedure, in total, about 0.5%, much smaller in the error frequency. So in the concluding remark, they wrote, we recommend that the high concentration protocol of Hikoya and Hayatsu be used to improve both the efficiency and the reliability. So we are quite happy to see this. Now this is in this slide I add a cautionary note about hydroquinone. I see that in many protocols, people add hydroquinone in addition to this dilute bisulfite solution. In order to this, the purpose is to avoid the decay, possible decay of bisulfite. Why do they do this? Perhaps this is because of our original paper published in 1972. I came to Cambridge in 1971, very hot summer. I stayed in the MIT dormitory without any air conditioning at the time, and did the experiment in Golding's lab in MIT with postdoc Bob Miller, taking piece of DNA and treated it with 10 molar bisulfite, 10 millimolar bisulfite in this case, very diluted, and observed very quick double-strand breaks in these DNAs. And this takes place because of the aerobic oxidation of dilute bisulfite. During this aerobic oxidation, free radicals are formed to break the DNA. So very dilute hydroquinone was quite effective to prevent this DNA cleavage. And these people used hydroquinone very probably according to this unwanted results. But I myself found this was unnecessary and even wrong. So I say erroneous use. Because bisulfite breaks DNA only in dilute solutions, 10 millimolar. 1 molar, 2 molar, 3 molar, no breakage takes place. So it is no use, this one. Plus, 90 years ago already, hydroquinone is known to react with bisulfite. In this German journal, Berichte, of German Gesellschaft, Chemische Gesellschaft, they describe that 1 molecule of hydroquinone picks up 3 molecules of bisulfite. So I'm going to finish this. Bisulfite genomic sequencing is a nowadays very, very important technique, and I recommend that high concentration bisulfite be used for quick and accurate results. And in the final slide, I say acknowledgements to my collaborators, Dr. Swataya, Ida, Namishi, Shiraishi, and others. My tutorial given to me was given by great professors, Ochiai, Professor Ochiai, Professor Kita both from Tokyo University, Professor Sugimura, a former president of National Cancer Center Japan, and of course, Covid Corona. Thank you very much.